repeats cause symptoms like involuntary movements, problem with thinking and speaking, and many more. Unfortunately, there's not a treatment to treat this yet, but Mike and some people in the lab are working on it. But there are treatments to help manage your symptoms. So this summer, my summer experiment was to perform PCR to characterize number of CAG repeats that our HD model cell line contains. So we did PCR, we ran a gel electrophoresis, and we also did process of Sanger sequencing. So PCR protocol is basically 3 steps. First is denature. That's when double-stranded DNA templates are heated to separate the strands. The second step is like annealing, and that's when short molecules called primers bind to flanking regions of the target DNA. And the final step is extension. That's when DNA polymerase extends the 3 ends of each primers along the template strands. So our main goal was to determine the CAG repeat number line in our Huntington disease cell line, also known as HDHQ111. So our experiment, first we made our mixture using Primestar GXL reagents, and then we put it in the thermocycler for 40 to 50 minutes. The thermocycler basically like amplifies DNA. So afterwards, while our mixture is running in the thermocycler, we set up a gel using 2% agarose, and then afterwards, into the gel we insert the mixture. But before we do that, we have to add a purple dye. It basically helps you see the mixture that you're inserting to the gel better, and also it pulls down the mixture to the bottom of the well. Alongside with the mixture, we also insert a ladder. The ladder basically helps us see what number we get at the end, the number of bands. So after that, we just let the gel electrophoresis run for an hour or so. And DNA is negatively charged, so in this process, this machine basically pulls it from negative to positive. So after that, we take the gels out the electrophoresis, and then we put it inside this machine called the imager. Alongside the imager, there's like a computer connected to it. That's how we can tell if we got any bands or not. And we did actually get bands, so this is me using a light box to cut out the gel pieces, gel DNA bands. And we have to do this step very quickly because DNA tends to break really fast, and UV light is also really bad for you. Challenges and discoveries. So we had to redo our experiment 3 times due to no bands showing up. And finally on our 4th trial, bands showed up. So we cut out the gel pieces and sent it for Sanger sequencing for them to tell us the DNA sequence. So the numbers that are numbered from 1 to 5 is what we cut out and put them in like each individual tubes and send it for Sanger sequencing. So when we got our results back from Sanger sequencing, we found out that the number of repeats in our HDHQ11 cells was 109. That means they are indeed in the range of Huntington's disease. So this is just the repeats.
And so the process that Ray did, Sanger sequencing, I took Ray's samples and took it a little further and did a experiment sort of based off of Sanger sequencing, which the way Sanger sequencing works is it's similar to how Ray explained PCR, except you have these fluorescently labeled molecules or oligonucleotides that as your DNA is amplifying through Sanger sequencing, you pass it through the capillary, which is just a small little tube. A laser sort of reads the frequency of the fluorophore, and it can tell your DNA sequence. The process I did with Ray samples later is I took them for capillary electrophoresis, also known as fragment analysis, and determined the size of the repeats for both copies of the gene. So after performing fragment analysis on our HD repeat amplified DNA, we get sort of these peaks right here, and this is sort of referred to as a spectra. And we're looking at 2 things. Firstly, we're looking at the height of each peak, and so the highest peak would be sort of that length of repeat that occurs occurs most commonly and would be known as the main repeat. The next thing we're looking at is the number of peaks across, which would correlate with instability. And so as HD progresses and as Huntington's disease progresses, the CAG repeat in a person's DNA becomes more unstable and it can vary more often. And so what we found is in one copy we had 99 repeats in allele 1, and then in the other copy we had 107 to 109 repeats. Both would be well within Huntington's disease range.
So our conclusion, our HGHQ111 cells have 109 repeats. So this would mean if a child has 109 repeats, that's considered juvenile Huntington's. So what I got out of this program / impact, so I got to explore different types of career paths that I never even knew existed. I learned about different sides of science that we don't really learn at school. I got better at communicating and managing my time. I'm now more experienced and comfortable with using lab tools, and I met so many great people like Defina, who's an MD PhD, and Nancy, who works in the FDA. Basically, Mike set up a meeting for me to talk to them one-on-one, and it was really cool to for them to share their careers and all of that. And then my supervisor, Mike and Zach, who made this summer experience fun. There wasn't a time in the lab where I was like bored. So, and then of course Miss Michelle and Maya, who made this program possible. And yeah, thank you to all my new friends that I made this summer. I'll miss you guys.